Sorry, I was muted. Hi, everyone. Thank you for joining the second webinar in our three-part series on using in silico and in vitro approaches for next-generation risk assessment of potential respiratory toxicants. This series is being co-organized by the US Environmental Protection Agency, Beta Science Consortium International, Syngenta, and Unilever. My name is Maria Balthazar. I'm a science leader at Unilever, and I'll be co-moderating today's webinar with Iris Muller, who is a molecular biologist also at Unilever. Today we have three presentations showcasing how in silico and in vitro methods can be used to help risk assessment. First, we will hear from Nikaeta Sadekar of the Research Institute of Fragrance Materials, co-presenting with Grace Patlewicz of the EPA Office of Research and Development, and Jane Rose from Procter & Gamble. The second will be co-presented by Gino Scarano of the EPA Office of Chemical Safety and Pollution Prevention, and Shawn Anderson from International Flavor and Fragrances. The last presentation will be given by Vivek Patel and Holger Bersinger from the Institute for In Vitro Sciences. Before I introduce our first speaker, I would like to point out that the slides and a recording from today's webinar will be posted at the link on the screen shortly after the webinar. You can also register on the web page for the next webinar in this series, which is being held on June the 2nd, and watch the recording from the first webinar, which was held on May 19th. There will be time for questions for all three speakers at the end of the last presentation. Everyone is on mute, but you can type any questions in the question section within your GoToWebinar toolbar, which should be on the right-hand side of your screen. If you send in a question or comment, please indicate if it's for a specific speaker or for everyone. I now turn over to Gino Scarano for his introductory comment.
Thank you, Maria. My name is Gino Scarano, and I'm a senior advisor in the new chemicals division in the Office of Pollution Prevention and Toxics. EPA is proud to be a co-organizer of this three-part webinar series, along with PETA International Science Consortium, Syngenta, and Unilever. I want to thank the organizing committee for pulling this together, especially Andy and Amy from the PETA group. So welcome all, and I look forward to all three presentations and an invigorating discussion. Thank you.
Okay, so speaking first today will be Dr. Nikaeta Sadekar, Grace Patlewicz, and Jane Rose. Nikaeta is a scientist at the Research Institute of Fragrance Materials, the RIFM, leading the respiratory endpoint for safety assessment and research efforts. She's joined RIFM in 2018 and is engaged in various collaborations across the scientific community for developing RIFM's respiratory science. Grace is a research chemist at the US EPA. Her research interests are focused on developing and applying TTC read-across approaches for different regulatory purposes. She has now worked in industry and government agencies of both sides of the Atlantic. Jane is a principal scientist at Procter & Gamble within the Global Product Stewardship Human Safety Organization, where she develops inhalation risk assessments for personal and household care products. And she is also the leader of the PAG Inhalation Expert Team, where she works to advance animal alternative approaches to safety assessments.
Thank you so much for the introduction. Hello, everyone. So Threshold of Toxicological Concern is, or TTC, is a universal limit established by combining all the existing relevant endpoint-specific animal data for evaluating if the exposure of a chemical is safe. In this presentation, we will see a use case scenario for inhalation TTC from RIFM safety assessment standpoint as an effective animal alternative method, and also the current efforts in enhancing the science. Hello, I am Nikaeta, a scientist at RIFM leading the respiratory endpoint, and I'm joined by my colleagues today here, Dr. Grace Patlewicz from EPA and Dr. Jane Rose from P&G. The presentation is divided into three main parts. The first part is the background on the threshold of toxicological concern as a concept. The second part is the introduction to Carthew's inhalation TTC approach. Both of these parts will be covered by Dr. Jane Rose. And the last part, the case study for its application, will be covered in 2 phases. The early efforts at RIFM to enhance Carthew's inhalation TTC will be presented by me, and the introduction to our collaborative project between RIFM, EPA, P&G, and Fraunhofer will be presented by Dr. Grace Patlewicz. And now, without further ado, I'll have Jane continue with the presentation.
Great. So thanks, Nikaeta. This is Jane here. I'm going to start off with the introduction to the TTC approach, some background and history, and then talk a bit about the inhalation TTC. So as Nikaeta mentioned, the Threshold of Toxicological Concern, or TTC, is a concept that refers to the establishment of human exposure thresholds for chemicals below which there is no appreciable risk to human health. And making the best use of existing animal data, this TTC approach relies on past accumulated knowledge regarding distributions of potency for relevant classes of chemicals, for which quality toxicological data are available. And based on this knowledge, an estimate of probabilities of no adverse effects occurring for a substance of unknown toxicity can then be made with an understanding of exposure. So TTC can be applied as a substitute for substance-specific hazard information where there is an understanding that human exposure is low, as well as the concept can be applied to prioritizing substances for risk assessment, where there may be hundreds or perhaps thousands of substances that need to be considered. So next slide, Nikaeta. So a bit of history on TTC. I'm not going to go into a huge amount of detail here, but just to ground us all in what the 2 main tiers for TTC that are available right now. So on the systemic tox side, so for the gene tox cancer tier as well as the non-cancer tier. So originally the gene tox cancer tier for TTC was based on predicted tumor risk of 1 in a million, derived through an analysis of a cancer potency data set. For the non-cancer tiers, an important publication from Monroe in 1996 explored the relationship between structure and toxicity through evaluation of a large data set of 600 chemicals. That had been tested for a variety of systemic endpoints from primarily oral, well, all oral studies. Substances were assigned by one of 3 structural classes that were defined by a decision tree that was derived by Kramer in 1978. And distributions of NOELs were found to differ for 3 classes, revealing how structural classes have an important bearing on toxicological potency. And then in 2004, Cruz et al. Put together these cancer and non-cancer tiers into a tiered TTC approach that established several human exposure thresholds over 4 orders of magnitude. So these range from 0.15 micrograms per day up to 1,800 micrograms per day. And it's at that lowest threshold, the 0.15, where substances present for an alert for gene tox all the way up to the 3 Kramer structural classes, with 1,800 micrograms being the Kramer 1 threshold. It should be noted that these thresholds were established for lifetime exposures and by the oral route. So Nikaeta, if you head to the next slide, so analogous to these past approaches to derive TTC for systemic tox or for the cancer endpoint from oral studies, here we are looking to explore the utility of the TTC concept to derive limits that are health protective for inhalation exposures from a data set of repeat dose inhalation studies. And once established, we see utility in applying the inhalation TTC approach for an individual chemical assessment, or in the case of where risk-based decisions are needing to be made to prioritize thousands of chemicals that may lack substance-specific data. Next slide. So why develop an inhalation TTC approach? Well, one of the reasons is the availability of inhalation repeat dose studies to support substance-specific safety assessments is relatively limited versus the availability of tox data from other routes of exposure. So this slide is meant to illustrate that concept that of all of the repeated dose in animal studies that are available, the number of studies available from the inhalation route is limited, and even more limited are those studies that have evaluated local respiratory tract effects as well as systemic effects. In addition, with the limited inhalation studies available, the potential for doing read-across approaches is also somewhat limited. And with various regulatory constraints around generating new animal studies, generating new data is often not an option. So if we can establish an inhalation TTC approach in cases where human exposure is sufficiently low to support the safety of a chemical, then this could be a useful tool. In the interest of time, actually, I'm going to skip this slide and move on to the next one and dive into talking about the Carthu inhalation TTC approach that was established back in 2009, where systemic and portal of entry effects were evaluated from inhalation studies. So I'm going to provide a bit of background on how Carthu constructed his inhalation TTCs, and talk a bit about some of the opportunity areas to build upon the Carthu approach. So to derive inhalation TTCs based on systemic and respiratory tract portal of entry effects, Carthu built a data set of repeat dose inhalation studies for chemicals that he considered representative of industrial compounds, and primarily gathered that data from 4 publicly available sources. They included chemicals that were gases, aerosolized liquids, and several chemicals that were solid particles. They included repeat dose animal studies of 28 days and longer conducted in rodents, and distinguished between local and systemic effects within those studies and established points of departure for local versus systemic. And in the end, they narrowed down the data set to 92 chemicals. It should be noted that Carthu had a list of excluded chemicals or classes of chemicals based on specific toxicological endpoints. So for example, genotoxic carcinogens, heavy metals, organophosphates were excluded. And of special note, the exclusion of polymers from the data set because of the potential for persistence in the respiratory tract. So I want to raise one point of within this analysis by Carthu, we recognize 92 chemicals as a good start, but if we compare this 92 chemicals to other TTC data sets, it's relatively small. And so this is one of the areas that Nikaeta will talk through later in improving, in bolstering the number of chemicals that are in the data set. This has also the opportunity to increase the chemical space that is being evaluated by TTC that then could be reapplied to a broader set of unknown chemicals. Next slide. All right, so Carthu used the Kramer decision tree to classify chemicals based on the presence of specific structural features and on toxicological potency. The decision tree, the Kramer decision tree, as I mentioned, was developed in the 1970s to predict tox risk of chemicals based on systemic effects from studies from ingestion, and assigned chemicals into classes 1, 2, or 3 based on low, moderate, or serious toxicity, respectively. For the Carthew data set, there were a little more than half of the chemicals binned into Kramer class 3, with 40% in Kramer 1 and only 4 chemicals in Kramer class 2. So Nikaeta, if you go to the next slide. All right, so this figure shows the TTCs identified by Carthew to assess local respiratory tract and systemic effects from inhalation exposure. And you can see that he separately derived thresholds for local effects within the respiratory tract versus systemic. And because of the few chemicals in Kramer 2 did not derive thresholds for Kramer 2 chemicals. But so if we focus on the local TTC thresholds, these were in units of microgram per person per day, and these were derived from 5th percentile NOx based on local effects from the 92 studies that were evaluated. These were corrected for duration and normalized to human lung weights with appropriate uncertainty factors added to derive human protective thresholds. A similar approach was used to derive systemic inhalation TTC thresholds, again in microgram per person per day. From a distribution of NOx, the 5th percentile was taken based on systemic effects from the studies, and again, corrected for duration and normalized to human body weights and appropriate uncertainty factors added. So you can see the local and systemic thresholds being relatively similar between the two across the Kramer classes. Okay, Nikaeta, if you go to the next slide. So this slide is meant to illustrate the distribution of the no effect levels from the studies that Carthew evaluated. On the x-axis are the local inhalation NOx plotted, and the y-axis are the systemic inhalation NOELs. And so we can see a pretty good distribution across the different chemicals, with hydrogen peroxide being on the more potent end and pentafluoroethane being on the higher no effect levels. So Carthew built a conservative yet practical data set of chemicals. So if I go then to the next slide, just briefly to mention how might this inhalation TTC approach be applied to a chemical specific safety assessment. So as an example, we're showing here isoeugenol, which is a fragrance chemical, and it lacks specifically inhalation hazard data. And so there's an opportunity to assess this material using TTC. Importantly, we need to know what the human inhalation exposure is to this chemical, and that data has been gathered by RIFM gathered from industry members, and so we can quantify what that human exposure is and compare what the corresponding TTC threshold derived for a Kramer class 1 chemical from Carthew, which is 1.4 microgram per person per day. Compare that to what the inhalation exposure is and derive a margin of safety. Next slide shows just one additional example of Nikaeta, which is also a fragrance chemical. It is the Kramer 2 chemical, and as I mentioned previously, there were too few chemicals in the Carthew data set to derive TTC limits for Kramer 2, and so we default to Kramer 3 thresholds of 0.47 milligram per person per day. And again, comparing to a human inhalation exposure, we can derive a margin of safety. So from this illustration, we can see the utility of using an inhalation TTC approach to support inhalation exposures to these fragrance chemicals. I'm going to turn it over to Nikaeta now. She's going to talk about steps that we're taking to build upon the approach of Carthu, including increasing the number of chemicals in the data set, increasing the chemical space, which I've talked about previously, and also exploring whether there are other methods that we can use to bin chemicals based on toxicological potency instead of relying on the Kramer decision tree, which was derived for systemic tox evaluations and from the oral route of exposure. And at the end, determine whether we can derive inhalation TTC thresholds for local and systemic effects. So Nikaeta, to you.
Thank you, Jane. So as you can see, with inhalation exposure to fragrances being very low, we get the values from the RIFM model and members of our member companies. And the application as such of inhalation TTC concept towards their safety assessment is of strategic importance. This exposure-based weighting approach is applied only in the absence of any chemical-specific toxicity data. So we exhaust our literature search for target data. If we don't have that, then we look for suitable read-across analogs with suitable data. And if we don't have that, then we refer to inhalation TTC. And as seen from the efforts already published by Yang et al. In 2017 and Patel et al. In 2020, which highlight the attempts for supporting the existing Monroe's TTC approach from '96. RIFM started this project in collaboration with P&G to similarly support the Carthu's methodology and move beyond the fragrance space. So initially, the main objective was to, as Jane mentioned, increase the data set and the enhancement of chemical space for the inventory. We also later on started probing a better way to classify the inventory, which would be relevant for observing and evaluating local respiratory effects for safety assessment. And we were looking to do this by clearly separating the local and systemic effects NOACs from the studies. So we started off by gathering our data set with the two main goals, as mentioned, increasing the inventory and diversifying the chemical space. And we combined the data set from Carthu's 92 materials, from publicly available studies from Repto's database from Fraunhofer Institute. And also we searched for the ECHA database. We also combined the studies that were available from P&G and RIFM as well. And in our approach, we also incorporated the PhysChem properties for each of the compounds in the inventory. This was not the part of the Carthu's approach either. And with the similar filtering approach as seen in Carthu, we ended up with 246 compounds, which have repeated inhalation exposure data, repeated dose inhalation exposure data in rats and mice. And the 246 compounds do not include chemicals with genotoxic potential or heavy metals or nerve agents or organophosphates. So as per Carthu, this inventory was also classified using Kramer classification system. And as you can see from the cumulative frequency distribution graphs, for both local and systemic effect values, we see a clear distinction between Kramer class 1 and Kramer class 3 chemicals, although Kramer class 2 is overlapping between the two of them. And however, like apart from a small data set from Carthu's approach, it was also criticized for using a Kramer classification system for evaluating local respiratory effects. So as a part of the effort, we also looked at the possibility if site-specific effects can help highlight the responsible PhysChem properties that can be used to cluster the inventory for establishing local effects inhalation TTC limits. But the major limitation here we quickly realize is that often a lot of the inhalation exposure studies do not report observations for the entire respiratory tract, and therefore we are left with making greater assumptions about what those effects could be anticipated for the observations that were not there. Either it could be a missing data for upper respiratory region or the lower respiratory lower respiratory region. And therefore, at this point, we recognize that establishing a threshold for inhalation exposure-based local effects might go beyond just identifying the NOACs. And so we looked at all the other attempts that have been made, and they are available in published literature, where they have looked at different avenues for categorization of the inventory, with still no clear categorization achieved, with a lot of either overlaps or even making an extremely conservative move of binning the materials and defaulting them to a more toxic class. However, more recently, Patlewicz and Nelms published a paper in 2020. They used the aquatic mechanism of action profilers as proposed by Reith et al. In 2009. Now, Reith and the group looked at the acute inhalation and acute fish toxicity and found a good separation between the reactive categories and the baseline categories. Now, Patlewicz and Nelms used the mechanism of action profilers for acute aquatic toxicity and found that they can separate the substances and based on the inhalation exposure data and propose new TTC values. However, in the evaluation of that publication, we also realized that the local respiratory effects assessment will need a detailed analysis of the chemistry and the toxicological potency of the chemicals. And over here, we think that machine learning might help with moving forward in that aspect. So what is machine learning? In short, machine learning is the ability of the computers to learn without being explicitly programmed. Now, for Inhalation TTC project, we have used a combination of unsupervised and supervised machine learning methods to identify key descriptors to establish Inhalation TTC limits. So we looked at the inventory by dividing it into 2 clusters and all the way through to 7 or 8 clusters. And as you can see from the spread of the data points here, the inventory can be categorized in a better way between 3, 4, or 5 membered clusters. The categorization or the separation of the data kind of gets murkier if you go below 3 or even certainly above 5. So the classification analysis was carried out for 3, 4, and 5 clusters with the evaluation of accuracy and cross-validation scores. We started off this analysis with over 900 features for the chemicals in the inventory, and with constant refining of the approach, we were able to narrow down those key features to less than 10. Now, some of the analytical methods in this process were also observed to perform better than the others, with the cross-validation and accuracy scores of 80% or more. As such, the machine learning process has highlighted the following methods, about 7 or 8 of them, and 4 most important key features for the clusters, namely number of atoms, van der Waals volume, the shape of the molecule, and the size of the molecule. Now, at the moment, we have not looked at how these 4 features compare with the toxicological potency of the chemicals in the inventory. But before we dive on to brainstorming the next steps for this continuation of this project, I will hand over the presentation to Dr. Grace Patlewicz to talk about the next steps for enhancing inhalation TTC as an effective animal alternative technique. Grace, your cue.
Thank you. Thank you, Nikaeta. So you've heard already now that sort of both there's a strong interest in developing new TTC values for inhalation, that the data set isn't necessarily as extensive. And you've now just heard about work that has been done both by RIFM and to an extent by ourselves in terms of exploring ways in which we can sort of subcategorize chemicals into groups and develop these types of TTCs. But there's a number of still challenges that are left, and that was really sort of provoked and prompted a Cosmetics Europe's workshop that was held just last year in November. There were many participants that were attended, many that had published on inhalation TTC approaches, including Sylvia Escher from the Fraunhofer Institute, myself, RIFM colleagues that you've just heard, as well as a number of other experts in TTC and inhalation toxicity itself. We discussed a number of different topics, opportunities for expanding the database. The CARFU database isn't very extensive or very expansive, and obviously there are interest in expanding the scope of that to accommodate different types of chemicals and really sort of expand the diversity that we're addressing. We talked about ways to subcategorize chemicals based on their inhalation toxicity potency. We've seen one example here in terms of how we could use machine learning approaches. We used, you know, mode of action, aquatic, acute toxicity profiles. But one thing that we did find in the discussions is that, you know, sometimes the Kramer classes that have been used traditionally for systemic toxicity endpoints really weren't all that effective in separating potency for inhalation. There was a great interest in, you know, how to move the science forward, addressing a number of questions around, you know, what are the significant knowledge gaps that exist? What should be the inclusion and exclusion criteria for the inventory? What's the domain of use? And what dose metrics for TTC limits should be considered? But at the end of the workshop, I think what really sort of came about was, you know, there are a lot of pressing questions. How can we build on that and recognize and form new partnerships to collaborate together to address these research needs going forward? So next and final slide. You know, there really is next steps in terms of how we move forward, how we grow, and how we expand the databases. The Inhalation TTC project that RIFM has identified room for where we can grow and where we can expand, and there are a number of next steps that we can consider. We've expanded, or RIFM has expanded their collaboration to go beyond their current work by including other stakeholders such as ourselves, as well as the Fraunhofer Institute. We're looking to expand the data set and also to consider how we can harmonize it. There are many questions around what are the study types or the conditions, which studies should be included or not. How do we curate the data set to ensure quality? You know, some of the work that we had done in the Nelms and Patlewicz paper that Nikaeta had referenced, you know, we had done a programmatic statistical curation, but you know, we could really take advantage of the inhalation toxicology's expertise in terms of really honing down on study quality. And we also have a debate and discussion in terms of how we can broaden the diversity in chemical space. We've discussed and we're looking at ways in which we should identify other appropriate categorization approaches, machine learning versus structural rule sets, as well as how we might enfold and explore and incorporate the utility of other NAM data streams, such as high throughput screening or high throughput transcriptomics data. We want to analyze the spread of local and systemic risk values across these categories and use that to help identify the 5th percentile threshold values for both local and systemic effects from inhalation exposure. And that really is going to involve a detailed scrutiny of those 5th percentile studies. We hope with this new collaboration to publish the outcomes of the research project, both in terms of the analyses, but also to make that data set, this harmonized and curated data set that we intend to develop, make that publicly available. And with that, I'd like to thank you for all your attention, and the three of us will take any questions that you may have.
Thank you all. As I said at the beginning, we're going to take questions at the end. So we're going to now move along to our next presentation. Speaking next will be Dr. Shawn Anderson and Gino Scarano. Shawn currently serves as the Global Regulatory Strategy Lead for the Engineered Polysaccharides venture within IFF. During her career of 25 years with DuPont and more recently IFF, Shawn has delivered R&D innovations on driving technology commercialization across a diverse portfolio of businesses, including crop protection, microbial diagnostics, industrial biotechnology, and biomaterials, and has served in management roles for QA/QC, project and product management, strategic planning, R&D, and product stewardship and regulatory. Gino has been a toxicologist within US Environmental Protection Agency since 1998. He's currently a senior advisor in the New Chemicals Division in the Office of Pollution Prevention and Toxics. Within EPA, Gino has also worked with the Office of Pesticide Programs. Prior to joining the EPA, he spent 12 years working in hazard risk assessment in the public, private, and non-profit sectors. Over to you, Gino and Shawn.
Thank you, Maria. Is my screen all set?
Yes.
I'm assuming it is. Thank you very much. So Shawn and I will be presenting this second case study on poorly soluble low toxicity polymers, PSLT, under TSCA. We first must mention that the views in this presentation are those of Shawn and myself, as well as our colleagues who will be mentioned in the acknowledgements at the end. So these are the topics we'll be covering today. I will speak very briefly to TSCA reform, how the new chemicals program evaluates high molecular weight polymers at the US EPA, and how Section 4H of the amended TSCA has changed our world, reduction of testing on vertebrates, and will inform this IATA and future ones. And Shawn will go through the specifics of the case study, and then I will briefly go through the newer PSLT chemical category decision logic that we have developed with collaborators. The Toxic Substances Control Act, or TSCA, was originally enacted in 1976, and it's the nation's primary chemicals management law in the US. There were no changes to this law for 40 years, and then in June of 2016, the Frank-Lautenberg Chemical Safety Act for the 21st century was signed, and here's a picture of that signing by then President Obama. Unlike other chemical control laws, such as the pesticide law, the Federal Insecticide, Fungicide, and Rodenticide Act, TSCA does not have upfront toxicology data requirements. Now, prior to this amendment, prior to June of 16, certain new chemical substances were dropped from consideration for regulation during the 90-day review period. But what happened after Lautenberg is that EPA applies default approaches for those same types of substances, and in some cases, this has led to unreasonable risk findings. EPA uses, we use a variety of methods to assess potential risks of new chemical substances that come in the door, including the use of chemical categories or read across based on analogs. Our new chemical program regularly uses what we call the new chemical category document, which was last updated in 2010, and it has 56 chemical categories that look at chemical structure and eco and health hazard, as well as environmental fate in some cases. But chemical categories are developed to help submitters with understanding the potential hazard concerns that we here at EPA have for a particular category of chemicals. And the categories usually include specific chemical definitions, boundaries, analogs, and testing recommendations. For poorly soluble low toxicity polymers, that's one category, and we're concerned that they may pose an inhalation hazard to humans. In this slide, and one near the end of this presentation, we're going to describe the category, its boundaries, and the hazard concerns. Initially, the PSLTs The concern was for high molecular weight polymers with a number average molecular weight of greater than 10,000 Daltons that meet certain exemption criteria, which is what the E2 means, that refers to the regulation for polymer exemptions, and are manufactured or used as particles with sizes in the respirable range, about less than 10 microns. They may cause inhalation hazards, including lung overload, via specific mode of action, specifically the impairment of alveolar macrophage-mediated clearance, as identified in rat inhalation studies. Overload is defined in this presentation as when the exposure concentration is sufficiently high or the duration sufficiently long enough to overwhelm this alveolar macrophage-mediated clearance mechanism. So in this somewhat busy slide, we describe our default approach for evaluating high molecular weight polymers. The table on the top provides some details on the hazard information for two of the analogs that we have been using to represent this category. I did realize when I was pulling this together that the references are not listed, but these are papers by Mule et al. In 1990 and 1991. The bottom table shows one of our risk calculators, which is essentially an Excel spreadsheet embedded in all of our new chemical risk assessments. So just briefly on the top table, it shows that data on one of two analogs, which are used for read across to the poorly soluble low tox chemical, depending on the new chemical substance being evaluated. On the top is a styrene butyl methyl acrylate copolymer, which is the 9,000 toner, which was the product used, and the other is polyvinyl chloride powder. The study specifics, the point of departure, and the effect of concern are noted here. The numbers in red outline the PODs, and they show that the benchmark concentration was used for the toner, 2.5 milligrams per cubic meter, for significantly decreased macrophage and increased lymphocytes in the lavage fluid, and then the LOEC, or lowest observable effect concentration, of 3.3 mg per cubic meter for the polyvinyl chloride, for the chlorins as alveolar chlorins as the effect. Now, in the bottom table, that simply shows how we calculate the risk, the POD, the exposure values, and the uncertainty factors that are used to arrive at the calculated margin of exposure. And it's important to show that in this case, depending on which analog is used, the benchmark margin of exposure can either be 100 or 1,000. And of course, this is the benchmark MOE is a multiplication of the uncertainty factors. It's 10x for intra-species, 10x for inter-species, and either 1x if we use the benchmark concentration as we did in the toner, or 10x if the LOEC is used for the LOEC to NOEC extrapolation. So that's how you arrive at 100 or 1,000. And just on the bottom right, it shows that in whichever one is used, given the exposure considerations here, there would be risk for this calculation. So for this particular PMN, the case study that we're going to be talking about, EPA did use this default approach in its initial risk assessment in 2018 and found risk due to lung overload concerns. For findings of unreasonable risk, EPA is required to take risk management actions, such as development of a consent order or a test order with testing requirements and restrictions on manufacturing, processing, use, and disposal to address these unreasonable risks before the company can commence manufacturing or processing. This resulted in a significant new use rule being issued in 2019. And for this case, this is what we did. And I'm going to read or paraphrase some of the red portions that are highlighted in this slide. This is the final rule that was issued in April of 2019. So we had concerns for lung effects, and the conditions of use of the PMN needed to include the following protective measures. It can only be used for the use described in the pre-manufacture notice or the new chemical application. There can be no manufacturing, processing, or use with particles less than 10 microns. And anything other than the use specified and the particle size, a company has to submit what's called a significant new use notification, meaning that we have to review it if anyone wants to deviate from those requirements. And the other important thing on the right is what we call PUI, potentially useful information. So what information could be gathered by a submitter that would try to, that could change this rule. And in this case, the PUI identified was actually pulmonary effects testing and rat inhalation studies. So these NER requirements were consistent with the way EPA's hazard concerns and testing recommendations existed for the respirable, poorly soluble particulates category, which is in the new chemical category document that I mentioned earlier. So this was what we used at that time. And the portions of that category document, which was last updated in 2002, are presented here. And what is highlighted is simply that there are 3 issues that are highlighted. The particle size, it's limited to the mean measured aerodynamic diameter, and there were no weight percent cutoffs for the chemical substance, and that's important because these are particles, and what we're going to talk about are high molecular polymers. It's limited to effects on the lung, and in order to test out, it was limited to in vivo testing. So as I pointed out earlier, around this time, Lautenberg amendments to TSCA were enacted. I do want to say that this PMN case study came in the door in September of 2016. And so this law was passed in June of 2016. And here is the first subsection of section 4H of the law. And I'll just read it. The administrator shall reduce and replace to the extent practicable, scientifically justified, and consistent with the policy of this title, the use of vertebrate animals in the testing of chemical substances or mixtures. So now I'd like to turn the presentation over to Dr. Shawn Anderson from IFF to continue. Shawn?
Great, thank you, Gino. Thank you for providing excellent context for the case study and the regulatory environment at the time that we submitted the PMN for the substance. Before I dive into the details of the case study, do allow me to provide some additional context on the biochemical nature, the technology platform, and the commercial applications of the particular polysaccharide polymer in this case study, which really informed our regulatory compliance strategy. So what you see on this slide is an example or numerous examples of various polysaccharide polymers that are extracted from plant or algal or other biomass, and including key examples here of cellulose, which is a beta-1,4 linked polyglucose, and starch, which is an alpha-1,4 linked polyglucose. So very similar to the substance we're going to be discussing in detail here. These kinds of polysaccharide polymers that are naturally derived are widely used today to provide unique properties in a broad range of different industrial or technical applications. However, the functionality of the polymers and its uses are often limited to the polymer substance as produced in the raw materials and by the processes that are used to extract these substances from complex mixtures found in the natural feedstock sources. Next slide, please, Gino. So IFF has developed a platform, technology platform for the production of polysaccharides, such as the alpha-1,3 glucan as shown here. It's produced from sucrose by an enzymatic polymerization process. Enzymatic polymerization is emerging as a technology platform to produce nature-inspired polymeric materials that are biodegradable, made from renewable non-fossil fuel feedstocks, and which can be produced with the design flexibility of synthetic polymers. The ability to produce alpha-1,3-glucan at high yield on an industrial scale by the enzymatic polymerization process has facilitated our exploration of the polymer properties and potential applications. In nature, alpha-1,3-glucan is one of the structural polysaccharides commonly found in fungal cell walls, including in those of many commonly edible fungi and bakers' and brewers' yeast, used for millennia in various fermentation applications. A similar polymer made of alpha-1,3 linked glucosyl units is also produced in the human oral cavity as the result of action of enzymes produced and secreted by members of the oral microbiome. So it's a polymer that we are frequently and very closely exposed to. Alpha-1,3 is a highly functional polymer that can be used in a wide range of applications, as illustrated here, including fiber, composite paper, and coatings applications, among others. And designed polysaccharides like alpha-1,3-glucan can be engineered to provide novel functionality, and when they are biodegradable like alpha-1,3-glucan, they have the potential for an improved in sustainability profiles that address a lot of the societal and environmental impacts often associated with synthetic polymers. As typically produced by IFF, the alpha-1,3-glucan is a high molecular weight polymer of about 140 kilodaltons, and it is water insoluble. Dried to a white powder with approximately 88 weight% solids and the balance water, it has a median particle size as produced of about 150 microns and no particles less than 50 microns in size, and as such would not be considered a respirable substance. However, for many of the customer applications and to fit into customers' manufacturing equipment or processes, IFF desired the ability to produce size-reduced particles of alpha-1,3-glucan with the nominal mean particle size of 40 or 17 microns. This would be accomplished by post-production processing to grind the polymer. The ground polymer processed forms of alpha-1,3-glucan contain respirable particles then less than 10 microns in size and are in the range of human respirable sizes. And that's what, as Gino mentioned, was triggering the determination by the EPA of an unreasonable risk due to potential for respiratory inhalation and the issue of the SNR that restricted uses that result in particles smaller than 10 microns in size. IFF provided to the agency considerable data demonstrating low occupational exposure potential with the use of standard engineering controls and personal protective equipment when working with the ground material. And also notably in most of the intended applications, the polymer is used as a filler or an additive, typically irreversibly incorporated into a matrix of other substances or engineered into fibers or coatings or articles in such a way that the polymer is not present in the final, presented to the consumer in a particulate or inhalable form. The polysaccharide, therefore, does not pose either an occupational or consumer exposure risk. Unlike polysaccharides obtained from natural feedstocks, the alpha-1,3 polysaccharide polymer is synthesized from refined starting material of crystalline sucrose or from sugar beet or sugar cane, so in other words, table sugar, using a well-characterized enzyme catalyst that has been examined quite extensively from a toxicological perspective, including Ames, chromosomal aberration, and 90-day oral tox studies, and independently assessed as GRAS. This enzyme catalyst is used to produce the alpha-1,3-glucan in a way that is well defined, of known composition and high purity, and is not predicted to be toxicologically active. We would therefore argue that the polymer does not meet the ethical standard for animal welfare necessary for the conduct of animal studies. So we embarked then on a weight of evidence approach to evaluate the potential for the polymer to produce lung overload. And that's what I will describe in the coming slides. Next slide, Gino, please. Within this framework, we considered in chemical, in silico, and in vitro alternatives to in vivo testing for animal welfare purposes. So we used a multifaceted approach to try and address EPA's concerns and their risk assessment. We provided the EPA with exposure monitoring results for production of the polymer, including the grinding operation and demonstrating low occupational exposure with standard engineering controls and PPE. We demonstrated that there was no reduction in the particle size with shipment of the polymer cross-country, so ruling out that as a concern. We re-examined alpha-1,3 glucan solubility using a simulated epithelial lung fluid as described in the manuscript by Boysa et al. And we worked with applied research associates to apply their multiple path particle dosimetry or MPPD model to predict distribution, clearance, and lung burden over a simulated long-term exposure to the polymer. We did also explore in vitro alternatives for characterization of endpoints. However, due to the many challenges that go into selection and qualification of in vitro tests and the more rapid progress that we made on the other approaches, we ultimately did not continue to pursue the in vitro study approaches. So for the remainder of my talk, I will focus on the in-chemical study of biosolubility and the in silico MPPD modeling and how these results were used to inform EPA's revised risk assessment determination for the alpha-1,3 glucan. So also shared on this slide are the results of some of the baseline characterization performed on a sample of the IFF polymer that was intentionally ground to mimic the smaller size mode for use in some customer applications. So it was ground to a nominal 18 micron particle size. And the test substance was fully characterized, as you can see in the upper middle plot with regard to particle size distribution, and compared to log normal distributions for key reference substances. Of note, the Xerox toner that Gino mentioned in the key publication by Mueller that is cited by the EPA and used as an analog in their assessment of concerns around the polymer lung overload category, and also in comparison to silica. As evident in the upper middle plot, the particle size distribution for glucan has a major peak and a shoulder of smaller sized particles. The MPPD model can model a particle size distribution with a single median and GSD, or as a bimodal distribution with two median diameters and GSDs, one for each mode. And that was used, as shown here in the bottom middle, to characterize the particle size distribution for the alpha-1,3 glucan. So this was modeled then as a bimodal size distribution with a smaller diameter mode at 5 microns and a larger diameter mode at 18.8 microns. These then were used as parameters to input into the MPPD model to estimate the deposition and clearance. In the upper right of relevance to the solubility determination study, there were no particles less than 0.2 microns in size, and this is relevant as we did a 0.2 micron filtration step as part of recovery of the polymer for ultimately gravimetric analysis and determination of solubility. Additional polymer characteristics were including density, particle shape, were characterized for both unground and ground polymer, and charge were also determined as shown in the lower right. The results here and on the following slides are all described and discussed in detail in a recent publication by Ladix et al. In Chemical Research in Toxicology at the reference and link provided here. Next slide, please, Gino. Okay, so we employed an in-chemical experimental protocol adapted from Boyce et al. To assess the bioaccessibility and biosolubility of the alpha-1,3 glucan. Two solutions that simulate epithelial lung fluid were prepared, the original Gambles and simulated epithelial lung fluid, or SELF, and then water was used as a control. While Gamble's solution is considered the pioneering synthetic lung fluid, this initial formulation has since been modified by several researchers. Boyce et al. In their review of published literature noted that inorganic salts, surfactant, lipids, proteins, organic acids are all native to the epithelial lung fluid. While many of these ingredients are absent from the original Gamble formula, they are contained in the SELF formula as proposed by Boyce et al. So we prepared these solutions and conducted the study as described in Boyce. And as I mentioned earlier, we prepared the samples at various time points following incubation at 37 degrees and processed the samples by filtration with 0.2 micron filters, took these to dryness, and then determined the retained mass gravimetrically. According to IUPAC and US Pharmacopoeia, a substance is said to be soluble if more than 0.1 gram of that substance dissolves in 100 mLs of solvent. If less than 0.1 gram dissolves per 100 ml, the substance is said to be sparingly soluble. And while alpha-1,3 glucan was not soluble in water or in original Gambles, a weight loss of greater than 0.1 gram per 100 ml of self-solvent was observed, indicating that the alpha-1,3 glucan is biosoluble and bioaccessible. Next slide, please, Gino. So we extended this biosolubility study by the application of a simple exponential decay model to the 10-day test data set to predict solubility in the self fluid using the relationship and equation as shown here. Based on the 10-day data set, we calculated a decay rate, or lambda D, of 0.0105 per day. This model was validated by then comparing its prediction with a second set of samples of the polysaccharide in self solution incubated for 30 days. Using this estimated decay rate based on the 10-day data, the exponential decay model predicted a 20% of the polymer would be dissolved after 30 days. And this agrees exceptionally well with the experimental observations that were shared in the table on the previous slide, where at day 30, 26.95% of the solution was in, or of the polymer was solubilized. Based on this model, we calculated the half-life of the IFF glucan to be 66 days, at which time 50% of the polymer is dissolved in the self-lung fluid. Next slide, please, Gino. Okay, turning now to the multiple path particle dosimetry, or MPPD, in silico modeling. This was as developed by Applied Research Associates, and here we were working with Owen Price from ARA to use this model to predict the lung burden for the alpha-1,3 glucan. MPPD was validated with measurements in rats exposed to the toner used in the Mueller study and as cited previously, and it showed good agreement with lung burden measurements as reported by Mueller et al. We modified the model to include a biosolubility factor for derived for the alpha-1,3-glucan polymer. The model takes the form of the equation shown here, where lambda A is the pulmonary clearance rate, and where there are fitted coefficients A, B, C, and D. M sub A is the lung burden in the alveolar pathways, and lambda D is an additional factor obtained from the biosolubility experiments that accounts for the independent mechanism of dissolution of the alpha-1,3-glucan. Data for amorphous silica were used to validate the clearance model, and the model incorporating dissolution predicted amorphous silica lung burden within 20% of the measured values, published values. Using the modified MPPD model then, a simulated 24-month rat exposure was run, and it yielded 10 to 20 times less lung burden for the alpha-1,3-glucan as compared to the toner at equivalent exposure concentrations. Next slide, please, Gino. So pulling it all together, the human equivalent concentrations, or HECs, were calculated for each toner rat exposure concentration, normalizing for particle size and differences in lung surface area. HECs were also determined for the alpha-1,3-glucan, taking into account solubility at the exposure concentrations, yielding the same predicted internal doses as the toner. These HECs represent the concentrations necessary for a lifetime occupational exposure to yield a predicted surface area normalized lung burden that's equivalent to the surface area normalized lung burden observed in the rats exposed to concentrations of 1, 2.5, 4.1, and 16 mg per cubic meter over the course of the 2-year Xerox toner study. Of note, the HECs for the alpha-1,3-glucan polysaccharide were over 100 times larger than those for the toner. If you look at the data highlighted in the red box and compare those values, this indicates a significantly higher safety margin for the alpha-1,3-glucan. Next slide, please, Gino. So upon a review of these data by the EPA, the agency considered the IFF biosolubility testing actually as a conservative and worst case estimate. And they further took into consideration the daily turnover of the intraluminal volume of the respiratory tract fluid. And for those of you that have interest, I would really recommend reading through the section from the proposed rule change that were abstracted here for the details of how the EPA actually took the data that we provided into consideration and used this to revise their determination. So basically extrapolating from the in vitro measures to in vivo, the agency actually determined that an effective predicted loading concentration that was 5 times lower than the loading concentration actually tested in the biosolubility study. Based on this weight of evidence approach, the EPA determined that the alpha-1,3-glucan polymer would not overload the lungs at the predicted worker exposure levels and concluded there was no need to conduct animal studies. As a result, the EPA revoked the SNR, restricting manufacture and uses of the IFF glucan polymer, effective September 24th of 2020. The case for alpha-1,3-glucan provides an illustrative example that MPPD may be an effective model for evaluating the lung overload potential of other polymers, and that selection of an appropriate lung fluid simulant may have a significant impact on the solubility determinations and predicted behavior then of a substance in the human respiratory system. In conclusion, then, it's therefore important to consider in silico, in chemical, as well as in vitro alternatives to in vivo testing for animal welfare purposes. Based on the IFF experiences, such methodologies may be utilized to build a weight of evidence and to potentially waive the need for animal testing when evaluating the potential of a polymer to produce lung overload, and further that non-animal testing data may be used to justify lifting chemical rules. So with that, I'd like to turn it back over to Gino to go through the next few slides.
Thank you, Shawn. And first, apologies. I shut down my email pop-ups, but clearly I forgot to do the chat and I couldn't do it. So I apologize for those who have seen some annoying things that I tried to control quickly. This slide simply shows a screenshot of the Federal Register notice that issued the final rule. And Shawn, thank you for showing the proposed rule, showing the rationale for making the change. And this just shows the final rule that went out, and it became effective in September, 1 month later. So there we go. So, oh, excuse me. So the IFF contributions did lay the groundwork for further refinements to the way we were looking at high molecular weight polymers. And so by some of my colleagues at EPA collaborating with external partners, including NGOs and members of industry, to further determine the breadth of non-animal test methods that could be used that provide information of equivalent or better scientific quality and relevance in accordance with Section 4H of TSCA. This collaboration led to refinements to the high molecular weight polymer category and the inclusion or exclusion criteria, which removed the molecular weight cutoff and included some respirability, solubility, reactivity, and biosolubility endpoints. So with this final slide, it shows the decision logic for the draft PSLT polymer, poorly soluble, low toxicity polymer categories. This was presented as a poster at this year's SOT meeting by a number of collaborators from EPA, industry, and NGOs. And it represents the evolution from the use of the existing category, poorly soluble particulates, and the default approaches through a several-year period of proposing and refining a new lung overload subcategory for polymers. So in this case, if you follow from left to right, this logic train, when evaluating inhaled agents, the PChem physical chemical properties are the major determinants for potential toxicity. So the polymers can be defined as those new chemical substances that have properties and that have the right properties and meet the functional criteria for these inclusion-exclusion properties. The first consideration, of course, is whether it's a polymer and then whether the particle size is respirable. Some may require evaluation for other hazard concerns. In other words, if you have an adverse effect resulting from like direct translocation of ultrafine particles directly to the brain. But the additional considerations going from left to right are whether the aerosol would be water extractable, bioreactive, or biosoluble. If none of those properties apply, then the candidate polymer would be considered a poorly soluble low toxicity polymer, or essentially a member of this category. So I'm going to give it back to Shawn to go through the final slides, and there you go.
Great, thanks, Gino. Well, both Gino and I just wanted to acknowledge some of our colleagues from IFF, my colleagues involved in the case study for alpha-1,3-glucan, and Owen Price from Applied Research Associates, and as well the folks mentioned there from the EPA who were involved in evaluation and review and discourse on the case study. And we'd be remiss not to also mention all of the external collaborators and that have participated in the discussion of the new decision tree that Gino shared in one of the previous slides. So with that, I think we will, on behalf of Gino and myself, thank everyone and thank the organizers for giving us this opportunity to participate in this very informative forum.
Thank you very much both for the excellent presentations. As a reminder, we will hold all questions to the end, but please send them through in the questions panel. So our last presentation today is from Dr. Holger Bersing and Vivek Patel. Vivek is a toxicologist and study director at the Institute for In Vitro Sciences. He leads the respiratory in vitro method and assay optimization and development projects and sponsor-initiated studies. Holger is the director of the respiratory toxicology at IVS. He's obtained his PhD in pharmacology and toxicology at UC Davis and has worked with various in vitro and ex vivo models for over 20 years. His current efforts are focused on optimizing human-relevant pulmonary models and developing technology to detect key events associated with lung disease progression. So over to you, Vivek.
Great, thank you very much. I am seeing if I can share my screen, or maybe it's already up. Is that right? I'm sorry?
Well, it's at the moment it says presenter as Vivek, so.
Okay, if we can move it over, give me a presentation mode, that'd be great.
Are we seeing now?
Okay, we have Vivek's screen. So Vivek, if you could just transfer control to me, that would be great, and I can go ahead and begin.
So now, sorry, Holger, now you're as presenter, so you could go and share your screen. Okay, good.
Okay, I think you can see it now, is that right? Or
Yes, yes, we can.
Okay, well, thank you very much for the opportunity that you gave Vivek and I to speak at this in this meeting session. And so today we'll be talking about three-dimensional in vitro models to predict relevant toxicity, but we also want to indicate, pharmacological effect. That's something we've been approached by in terms of some of the sponsors that have come to us and asked about some of the different test systems. They want to look at certainly the potential toxicities of their materials, but also pharmacological efficacy. So we're very pleased that these test systems are gaining more traction. So let me see if I can get, there we go. Here is a brief agenda. I'll start with just one slide about IIVS, who we are and what we do. I think many of you are aware of IIVS, but some of you aren't. So we'll give you a snapshot of how we fit into the whole in vitro world. Briefly talk about inhalation exposures and methods. We've heard a lot about that already, and so I'll just touch on a few of those points again and how that applies to the approaches that we take at IIVS. Then really focus on the test systems and endpoints. I'll briefly go over reconstructed human airways, just touch on human precision cut lung sizes, and then I'll hand it over to my colleague, Dr. Vivek Patel, who will talk about advancing human precision cut lung sizes. It's really kind of a case study of how you can take a test system that hasn't been that accessible to many laboratories and changing that. So IIDS was founded as a nonprofit almost 25 years ago to promote the use and acceptance of in vitro methods. And we provide testing services to a variety of different industries, including cosmetic household products and others, including the pharmaceutical sponsors that we're now seeing approach us. And we'd like to use our knowledge to create education and outreach programs and promote the regulatory acceptance of these methods worldwide. We've had staff going to China, to Europe, to South America, really not just educating others about in vitro methods, but actually teaching them how to use them. So we're considered an independent technical authority. We have science, education, and outreach, kind of a three-pronged approach to promoting non-animal testing. So we've heard about a variety of different materials from the other speakers, and we certainly are approached by different industries that have household products, personal care products, occupational products, other products. We've had large tobacco companies saying we've got these e-cigarettes that we like to test as well. And we're certainly happy to apply the non-animal test systems to that end as well. And so you can see here, with all these different types of materials and different sizes that we know have a large impact on where there's an exposure, we take that to heart and we work with the sponsors to come up with a reasonable approach. We've seen this probably a few different ways already from previous speakers, either a week ago or earlier today, that we know that the size matters. And so it's really only the smallest particles that are going to be making it down to the alveolar spaces, with the larger materials being really only accessible to the nasal cavity and perhaps the trachea. And there's a number of different deposition mechanisms. We had speakers, I believe it was last week, much more versed than I, talking about the different mechanisms that can impact the exposure in the different regions of the human respiratory tract. Well, as I mentioned, we are approached by different industries, and they have different types of materials. And so, we've come up with a way that we can come up with a reasonable starting exposure, because we have some sophisticated exposure equipment, and we can make exposure estimates, but we want to start with a reasonable exposure paradigm for the sponsor. We'll ask about particle aerosol size, atmospheric concentration, density, solubility, some of these different factors that you've already heard about, and how they can impact the amount of exposure that different parts of the lungs receive. We have sponsors coming and said, we've got a greenhouse environment. It's this many, cubic meters or square feet, and different ventilation rates. We have workers that may be in a room for up to 8 hours, and so we really ask for everything that they know about their material, and we take all that and we put it through some calculations to come up with that reasonable starting dose, and then we might add a higher dose and a lower dose, kind of flanking what we believe would be a reasonable exposure. But we're not doing this on our own. We have been applying some of those really nice references that are out there that you've already heard about earlier, by Rick Corley, computational fluid dynamics, and Bauman Asgharian with the NPPD modeling, and certainly we work with the sponsors and make sure that we come up with a strategy that works for them. Well, once we know what kind of an exposure we'd like to create, what concentration, we have different options. And I'm not going to go into these in great detail. But when people think of in vitro models and testing, you're probably thinking about an aqueous exposure. You take that test material and you solubilize it in a physiological buffer or a medium, and then you pipette that on top of maybe two-dimensional cells that you'd have adherent to the bottom of this polystyrene multi-well plate. And so that's probably the easiest, certainly the most economical. You can even use tissue culture inserts, which you would have hanging. You can do an exposure to these cell layers that are on top of the membrane, or you could conduct an exposure through the basolateral compartment where you have diffusion of that material through that porous membrane and affect the tissue from below, or you can do both, and then you can affect that exposure right away. Of course, with an aqueous exposure, you need to have test materials that are aqueous soluble, and that is seemingly more often a challenge for the test materials that we are approached to test for our sponsors. Well, we have another device. It's our HP D300. It's a digital dispenser, something I've talked about in other presentations I've given. And really just think of an inkjet printer that can take a DMSO-based material and deliver extremely low volume droplets to a multi-well plate. You can do a 1 to 1,000 dilution directly into a plate that already contains cells, or they came up with a biopattern printing application. And that's where you can print an array of these droplets. And here you can see in this picture here, these are 3 nanoliter droplets. These are really large. They were designed to be large, so you can actually see them with a naked eye. And with the tissue insert you see here on the right, you can see we successfully delivered accurate pattern to the top of that tissue culture membrane. And down below, we used not a red food color tracer like above, but a fluorescein tracer, and we delivered directly to the mucus layer of one of those 3-dimensional culture inserts. In fact, it was a mucillar tissue, and those tissues are advanced. They have ciliary beating, and so all those beating cilia just mixed up that exposure in a really nice homogeneous exposure. So if it's not aqueous soluble, this is certainly an option. DMSO is very commonly used for materials that are poorly soluble. But we also have our aerosol exposure devices, our Vitrocell Cloud 12 and Vitrocell VC1 with the help of PETA funding, so we're very thankful for that. And so the Vitrocell VC1, it's really a smoking robot, kind of an entry-level smoking robot. It has a single port, but it allows us to control very, very tightly the puff parameters. How long is that inhalation event? How long is that expulsion event? And then, of course, we have our exposure chambers as well, where we have our tissues contained. Here on the left, we have the Vitrocell Cloud 12, and this is gaining a lot more traction recently. We have sponsors coming with cleaning agents, really non-combusted aerosol exposures is what they'd like to create. And so that is, we have a number of different options really depending on that test material type. In fact, the Cloud 12, even though you typically would start with an aqueous-based solution, we've successfully dispensed quartz silica, fine ground quartz silica, even waterproofing agents kind of make an emulsion with an aqueous solution. We're able to deliver to the tissues that are contained here at the bottom of that exposure device. Well, we're here to talk about 3-dimensional test systems, but I wanted to show this one slide about 2D respiratory cell cultures, just to really contrast what's out there, 2D versus 3D. And so there are some cell lines that are derived from the respiratory tract. There are A549, these are cancer cells. H292 and the Bs2B, these are all immortalized. And of course, there's primary cells as well. Here you have maybe a few doublings, and then the cell culture ends, and you can go back to that supplier, and hopefully there's some more cryovials of those primary cells if you want to work with that same donor again. But these 2D inherently has its limitations. It's really only one cell type. And so your responses may be more limited in lifespan, and so they are well suited for a screening paradigm. And so if a sponsor comes to us and say, I've got many different types of materials, we'd probably want to start with a 2D testing strategy and then move to the 3-dimensional testing strategy after that. So when we think of the 3-dimensional test systems, we have a lot of options, and this is very welcome. There's a number of different regions of the respiratory tract that we know receive different particulate sizes, different exposure levels, and so we've got really good representation. Our respiratory tox group at IAVS, we also do work with oral tissues when it's warranted. But of course, we have the nasal cavity, trachea, and bronchi. Those regions represented by epi-airway and the mucilary tissues that are out there. There's also small air available now, and also epialveolar. But one area that we'll be speaking to you more about today is precision cut lung sizes. So, this is something that that goes way back in my history. Been doing PCLS since about 2003, and we're thrilled that we have a really nice human precision cut lung size test system methodology developed at IIVS. So reconstructed human airways or reconstituted human airways, for those of you that aren't that familiar with them, they're grown at the air-liquid interface. I mean, you'll hear about organoids and spheroids, and they are essentially the same when it comes to cell composition. You have 3, 4, 5 cell types typically contained in these, but with the ALI cultures that are grown at the tissue culture insert, you have the advantage of being able to access the different compartments. So the apical compartment here is really akin to something like the lumen of an airway. And so you would have the ciliated cells oriented towards the top here. You can kind of see the cilia here. You have a mucus layer. We can sample this. We can do an apical rinse and look for cytokines. And we found that very often there's much more marker, whether it's a cytotoxicity leakage marker or something like a pro-inflammatory cytokines being present in that apical compartment when you have an apical exposure. And many other researchers are always just sampling the medium. In reality, we're finding that you can have a lot more marker in that apical compartment. So you have those compartments. You can do different things with these compartments. Very often we've been asked to do omics work. We'll farm that out to one of our alliances or contractors that we work with. And there's a myriad of different endpoints that we can look at in-house as well. Here's a table that we put together to kind of highlight what we can do at IIVS with these different test systems, because very often we get the question, okay, this is my problem, what do you advise? And so we'll respond by saying, well, what is it you're trying to accomplish? And here you can see when it comes to the different biomarkers and events, we really try to actually orientate these in a way where it shows, the molecular initiating events, the cellular key events, and then the more complex events, down below. And you can see those three-dimensional models, the 3D reconstituted human airways and PCLS. You'll see more of those boxes or more check marks down here below. And so this is something that we can share with our sponsors, and so they get a much better understanding of what can be done with the different test systems that we have available. Well, something that we're really excited about is that we have collaboration with RIFM now to look at respiratory sensitizers, and so we are working with human precision cut lung sizes. That's our test system of choice. Even though the reconstituted human airways, you've got a few different cell types there. Well, the precision cut lung sizes, you have all the cell types contained in the tissue at the time of slicing. That would include the dendritic cells that you can see here in this schematic, is implicated in the respiratory sensitization event, just as it is known to be involved with skin sensitization. And you can see there's a number of different processes here. And we know that these in vitro or ex vivo test systems they're closed systems, so you're not going to have recruitment of cells to the site of exposure or challenge. But we can at least look at some of these initial steps, which we believe to be part of the respiratory sensitization. And again, we believe it's going to be human precision cut lung sizes that are going to deliver the answers that we'd like to see. We like AOPs. This is extremely helpful. We were also commonly asked, well, how do you know that your in vitro model is giving you the effect that you would expect to see in vivo? It's like, well, what happens in vivo? If I were to ask an in vivo scientist, okay, you had a systemic exposure. I need to know how much of that test article made it to that organ of concern and for how long. And that's where things get very complicated. Having supported the NCI previously in my prior role prior to joining IIVS, you're looking at Cmax, the maximum concentration in plasma, or maybe area under the curve, but it's largely not that well known how much precisely of that test material actually got to the site that you're concerned about. And so we really like these AOPs. Once there's a key event that we know is part of a process, for example, here you can see some of those processes that we believe to be part of respiratory sensitization. You can see the dendritic cell component in there. That gives us a target to shoot for when it comes to our non-animal test systems, and we want to see if we can see changes in those particular markers in those key events that we know to be part of that disease process. I mentioned that PCLS have all the different cell types at the time of slicing, and here you can see macrophages. Here you can see macrophages on the left, where we have a control culture 72 hours, and on the right we have 5 microgram per mil lipopolysaccharide, which certainly created a change. And you can see how many more macrophages are actually lighting up. And I just said that's the closed system, so you might be asking, where did they come from? We believe they're already there. It's the challenge from that test material that actually triggers the activation and staining of these cells. And the pathologist that we work with, I've known him for probably about 17 years. He can distinguish between the intraalveolar component and interstitial component. We had a nice presentation just before ours here about lung overload, and I can't help but wonder if there's some way that we can use these slices to see if there's some kind of a prediction that we could make. So we have cytokines. We have a very robust cytokine response with slices. Here, we can monitor the effects of the quartz silica. Here we monitor them for 5 through 8 days, 9 through 12, and 13 through 16. We pooled the medium in these different segments, and you can see that we can look at what appears to be kind of akin to chronic inflammation. So we have these inflammatory markers elevated for a period of time, but it's not just pro-inflammatory cytokines, but there's a number of other markers that we can look at with our multiplexer. And so this, really tells us that we've got a really good tool set to look at some of the different changes we'd like to see, certainly those that are believed to be involved in respiratory sensitization. So with that, I'd like to give the presentation over to Dr. Vivek Patel. He's done a lot of fantastic work at IIVS, and he'll be telling you a little bit more about what we've been able to do with our human PCLS to make them more accessible. Thank you.
Thank you, Holger. Hello, everyone. On the next couple of slides, I will be mostly going over some data from IIVS's previous and ongoing projects that are mainly focused on long-term culture and cryopreservation of human PCLS. And okay, and the data you can see on this slide, the top panel, these data are from the fresh human PCLS that we cultured for up to 28 days, and we could see that the slices were viable up to the day 28, that is throughout the culture period, and we did not see any major drop in the viability as assessed by the WST8 viability assay, as well as histology scoring by our pathologist. So we also tried culturing a cryopreserved PCLS. We use IIVS developed cryopreservation buffer to freeze these slices, and we have also tried culturing these slices at different culture conditions, namely air-liquid interface as well as submerged. And you can see in the graph on the lower panel that the slides are still viable even after day 21 of thawing. And if you look at the histology micrograph on the right, the yellow arrows are pointing at the macrophages that were still seen at day 14 in culture. So basically, these data, these preliminary data tell us that the slices are still viable in culture even after freezing and thawing. And our next work was mostly focused on characterizing the cryopreserved PCLS and to see if we can maintain the viability and functionality of the slices in culture after thawing. This work was supported by PETA. So thank you for thanks PETA for that. And what we did here was we obtained a normal lung and generated the slices. Now, after the slices were either used fresh or used fresh and cultured for up to 4 weeks at air-liquid interface, or they were cryopreserved for and stored for approximately 7 weeks and then put into culture and maintained at ALI for up to 4 weeks. The figure over here, it shows the time points at which the slices were either treated with Triton-X 100 or LPS. Triton-X 100 is a known cytotoxic agent, and LPS is a known inducer of immune responses. And the time points over here, the ones indicated in blue, are the time points when the slices were treated with either Triton-X or LPS. And following 24 hours of the treatment, the tissues were assessed for various endpoints, which included viability assessment using WST8, cytotoxicity using LDH leakage as a marker, as well as various tissue responses using the Luminex multiplex assay. I'm sorry, it's just taking a while to change the slide. All right, here we go. So the graphs over here are showing you the viability and cytotoxicity data. The graph on the top left is the viability data, and as you can see, the viability was pretty much comparable throughout the culture period for both the fresh and cryopreserved PCLS, and we did not see any differences, any major differences in the vehicle or LPS treatment groups. However, when the slices were treated with Triton-X, we saw a significant reduction in the viability. And this viability data is very well associated with the cytotoxicity data, which shows that in the Triton-X treatment group, the LDH was released at significantly higher rate as compared to the vehicle and LPS treatment groups. The graph on the lower left is again the viability data, but only showing the slices which were not treated with Triton-X or LPS. So basically they were just cultured, and it has some additional time points at which the viability for was assessed. And you can see the viability was pretty much consistent. And we also looked at the total protein, indicating the biomass of these slices. So these data basically show that both fresh and cryopreserved PCLS are able to respond to a cytotoxic agent. And we see the responses among fresh and cryo PCLS are pretty much consistent. Next question was whether we can see the LPS-induced tissue responses in these slices. Now, in one of the previous slides, I did indicate that we saw the macrophages in the cryo-PCLS even at day 14 after thawing the slices. So we just wanted to see whether those cells are just sitting there being viable, or they can actually respond to any insult as well. And as you can see, the graphs on the top panel, they show that we observed LPS-induced IL-6, IL-8, and MCP-1 responses through the 4-week culture period, both in the fresh as well as the cryopreserved PCLS. And the graphs on the lower panel, TIMP1 and GROW alpha, even though we observed detectable levels of these biomarkers, we did not see a major response to LPS for these biomarkers. And again, as I indicated, this study is still an ongoing effort, and we are still exploring to get more consistency in the responses, and we are gonna work towards it next. Sorry about the delay. I'm still waiting for the slides to change. All right. The next efforts at IIVS, they are mainly focused on exploring the application of HPCLS, both fresh and cryopreserved, for test material screening. And some of these projects have been already initiated. As Holger mentioned before, we are working on identifying markers for respiratory sensitization, along with in collaboration with RIFM. The other projects include inducing fibrotic phenotype and testing some of the antifibrotic agents and see if the model will give us some good data. We would also like to make a comparison of these responses from the PCLS from different donors belonging to different demographics. And at the same time, our efforts are also underway to push forward the standardization of HPCLS culture methods, which would hopefully give us more consistent data across different laboratories, and will help positioning HPCLS as a test system for regulatory testing applications. And with that, I would like to thank everyone for their attention, and of course the IIVS staff. You can see them in the picture on the right for their great work, and also our Chief Scientific Advisor Frank Gerberich, and our pathologist Khalid Amin for their support throughout these projects. And with that, I am done over here, and I will be happy to take any questions.
Thanks, Vivek. Thanks, Holger. It's a really great presentation, and thanks everybody else for like the really, really fantastic presentations. So I think we have like something like 12 minutes left for the questions, and I will start with the questions for the first speakers, if this is okay, so people also can finalize putting their questions in for Holger and Vivek. First question then for the first talk, which is like actually two questions, which I think are quite similar. How well do historical animal experiments describe the local effects in the lung? Is there a potential that you are missing undescribed effects when setting the TTC? And related to that, can you please elaborate how did characterize local versus systemic effects after inhalation exposure? Are traditional OECD inhalation studies primarily focused on systemic effects? I'm not quite sure who wants to answer that.
I can, this is Nikaeta. Thank you for the questions. I can attempt, and I'll pull in my colleagues for any additional information. So I think if I can remember correctly, but the first part of the question asking how well do the local effects can be characterized from the historically from the animal studies, is that right?
Yeah, yeah.
So I think I highlighted on one of the slides that definitely we came across this limitation where not all the inhalation animal exposure studies look in a very detailed manner for the local effects via inhalation exposure. So either they focus on those areas of the respiratory tract that actually do show severe effects, and those are recorded and well documented, and no comment or mention of whether other regions in the respiratory tract showed any mild or minor effects or anything, unless if they are following OECD guidelines, which I think now I'm going into the second part of the question. Those studies that are following OECD guidelines with inhalation exposures, they are well documented, and so they do have good description on and analyses on the kind of effects observed within the respiratory tract, and that has become our go-to place to look for local effects and use them towards our evaluations. When it comes to building the data set for inhalation TTC, what we have already done with the RIFM PNG collaboration, we focused on those studies more, and we tried to weed out all those studies that we found were ambiguous in terms of if we had to make leaps of assumptions, for instance, about the kind of effects that were seen. Also, if the studies are not very detailed in terms of mentioning the regions of within the respiratory tract that were analyzed, whether it's just histopathology or even biochemistry, we tried to weed them out from the list as well. So I think I should say that considering the number of chemicals, which is at present, I mean, we ended up with 246. We started with a lot more than that, and even for each of those chemicals, there were instances with multiple studies with multiple durations. So it was actually not very often that case, having so many options to deal with. But wherever we did, we found that the year of publication was a good indicator, also whether or not they were they would be following anything closer to an OECD guideline. So we were looking for such details as we were formulating or formatting that inventory. And Jane and Grace, if you would like to add something to that, please go ahead.
Nothing from me, more Nikaeta, thanks.
Nothing further from me either. I think you did a great job answering that one.
Okay, so then I have the question if the data set of the 200 plus chemical is publicly available, and how were the toxicity data of these chemicals curated?
So for I think the first question is asking if we have the 246 chemicals made publicly available yet.
Yeah, I think so.
Yeah, not yet. I mean, this project was not completed before we moved on to the new collaboration. So but it will be made publicly available as part of the more ongoing, broader goal for this project now. So, but the curation of the data set, we evaluated each and every study, and we set the parameters which overlapped CAR-Ts a lot based on following whether or not the studies are following guidelines, the duration of exposures, preferentially being from chronic to subchronic to subacute. However, we found that for some of the chemicals, such as, for instance, some of the fragrance ingredients, there are animal exposure studies that are following OECD guidelines, however, but there are major adjustments or deviations in terms of the duration of exposure. So we included them to take them under consideration. But not necessarily that means that they were completely accepted to be included in the data set. So they were considered for evaluation and curation of the data set in terms of the chemistry as well as the quality of the data. definitely we're looking at inclusion and exclusion criteria as part of the forming the inventory as well. And I think I have one of my colleague, Grace, reminding me that polymers were the ones that we decided to exclude, particularly because of the biopersistence quality or the property of them. And we are still looking right now as a broader collaborative effort into diving deeper into the exclusion and inclusion criteria afresh. And so I mean, I'm sure we'll be having more updates with regards to a better curated approach for better curated data set, sorry. So yeah, I hope I answered the question. Please do help me remember if there was another part to that one.
No, I think you covered probably everything.
Okay.
Just due to time, there are more questions for you, but I think I move on to the talk number 2. And the first question goes to Gino. So thanks for the presentation. I have 2 questions for slide number 5. First, how did you identify polymer analogs? And second, how does the number of workers affect mode of action, MOE calculations? Margin of exposure calculations, sorry.
Thanks for the question. I remember the first one, and I can answer that quickly. I mean, there was a lit search done as I understand it, published literature, gray literature, and then literature in our CBI files. And of course, and then these were candidates that came forward that seemed to have the most robust data set for what we were interested in. And so that's how those analogs were identified, the toner and the PVC powder. And could you repeat the second question?
How does the number of workers affect MOE calculations?
The number of workers?
Yeah.
It doesn't. I mean, we do it. I mean, in my view, the whole idea is you compare the exposure and the hazard value to come up with your MOE. The number of workers then, which is often identified by the submitter, it goes into the risk management realm for determination of how that's important in making a final decision.
Okay.
While I have the floor, can I say one thing, if you don't mind?
Yeah, that's right.
On our last slide with the acknowledgements, I understand that this is recorded, so I did want to do this. We ended quickly, and I did want to make sure that Tala Henry, Todd Stediford, and William Irwin, they were the 3 that did pretty much all of this work. I just reported it. So I wanted to make sure people understood that. Thanks.
Thank you. So for the alpha-1,3 glucan case study, could the polymer affect the lung lining fluid in ways not related to overload?
I mean, I will probably leave that to Shawn, if that's okay.
Yeah. So, yeah, this is Shawn. Based on the our analysis of read across and to other similar polysaccharides, most of the concern comes from those substances from inhalation of the other components that may be part and parcel of the preparation as they're coming from the mixture. So example, like wood dust is basically cellulose, but it's actually very complex, lignocellulose, lots of other constituents, potential contaminants, things like that. And it's often those other components that are actually the issues. And so it was in the case of the polysaccharide polymer, we're making this then under very defined conditions. The main other component are, as residuals, could be from the enzyme catalyst, and that's why we went through extensive measures to characterize the toxicity potential for those. And we do know that enzymes can be respiratory sensitizers, and that's why we also take measures to keep the levels in the final preparations as low as possible and below levels that have otherwise been identified as being concerning for inhalation of enzymes.
Thanks a lot for that. So I'm really sorry, but you want to add something? Sorry. Yeah.
Yes, I did. I apologize. Another mea culpa because I wasn't intimately involved. Annie Jerabek, whose name is not even on the slide, was a major contributor to this from EPA. So apologies.
Okay. Then I think I move on to the last talk just due to the time. For Vivek, how are the human donors selected for PCLS? What are the criteria for that?
So it was mostly non-diseased lungs that we accept, and these lungs are mostly not usable for any transplants due to some issues with them. But we don't have any specific criteria. We just try to make sure that the lung is still in good condition that we can use to generate the slices. Holger, do you think if you can add anything to this?
Yeah, it's extremely complex in terms of the inclusion criteria, and as Vivek mentioned, you know, the best lungs go for transplantation as they should, but we're looking for things like inflammation, pneumothorax. You know, there's a number of other clinical events that would actually exclude the lungs. You know, certainly if the sputum or bronchial swabs are positive for cultures, we don't want that. We want to be able to culture them for a period of time. So, you know, each lung is different, but we definitely want our lungs to have the best chance of success. We also acclimate our tissues or our slices for a number of days to, you know, if there is any residual effect just from the organ removal and shipping and all of that, for those tissues to acclimate before we use them.
In regards to that, there are two questions which are quite related, and I think this goes to donor variability. Do you investigate it in that, and did you see something like that?
This is an area that that's really now possible to assess. You know, with the cryo PCLS, we can start banking these. I mean, we currently have two healthy donor lung slice batches banked away in our liquid nitrogen freezer, as well as 2 COPD lungs. So now that we can start banking these, we can wait to get an assembly of different donors and then put them all through the same test at the same time. And so that's a question I get all the time, and one of the reasons that we've done this cryopreservation effort is not just to make these slices readily available to, you know, we can begin testing in 2 days if we were to have a study in place. And but now we can start looking at differences in response due to donor demographics.
Thanks a lot. So we already overrun a little bit of the time, so I think we end it here now. I want to thank everybody again for the great talks and for joining today's webinar. And I also want to make sure that there's still time to register for our next week's webinar at the 2nd of June. And enjoy the rest of your day. Bye. Thank you.
Thank you.